This conference will now be recorded.
I think there was some server problem. That is why the meeting has ended. Okay. Mansa, Priyama, you are getting the things? Both of you. You are getting what we are discussing? Write down the questions. Okay, write down the question and write down the answer in just one word. Write down the answer. Fine. You will be getting the PPT, so at that time you can read, but just write down the question. And again, I'm telling you, make unit wise, make the books. It will be very helpful for you at the end of the course. Okay, make different books, different, at least separate your books and make into different units, unit 1, unit 2, unit 3. So that will be helpful for all of you. Okay, so what I was telling is that, so what is the pH of the lysosome? The pH is acidic. And what is the number of that pH? That is about 5 to 5.5. So just now I was telling you the marker. So the next question is, what is the marker of the lysosome? The marker name is acid hydrolases. Just one word, acid hydrolases. If the environment is acidic, that is mostly it is lysosome. And suppose if any organelle is having this acid hydrolases, the probability is the organelle is lysosome. So the marker is acid hydrolases. Fine. And I was telling you that why this is having an acidic pH, because of this proton pump. Okay, because of this proton pump, it is having a acidic pH. And this proton pump is an active receptor, where when the ATP is broken down to ADP, H plus will move inside, and this H plus will create an acidic environment in the lysosome. And see this is acid hydrolase, an enzyme which can only survive, only be active in this acidic environment. Okay, and which is that organelle? That organelle is your lysosome. So what is the important question here? The acid hydrolases, the pH, the double protection, what does that mean? Here the 4 marks analysis can be asked. Then what is the what maintains this acidic pH, and what is this proton pump? That's it. So let's see what is the function of your, is there any other function of acid hydrolases? Yes, acid hydrolases, it will hydrolyze everything. Okay, it is one of the factor, one of the enzyme which is useful in the degradation pathway, in degrading the stuffs. Okay, and it will give you this acidic environment, and it will ensure that whatever is coming in this lysosome, it will be, it will be just broken down, and it will be like it will be finished. Because other things, if you are talking other organelles, acidic environment cannot work. Okay, so there are 3 major pathways. Okay, the first pathway, see here, the first pathway is called as the phagocytic pathway. What is phagocytosis? Phagocytosis means eating. In very simple word, you are eating something, and you are eating something which is solid. Okay, so this is the solid thing which you are eating, a bacteria, a foreign particle, a virus, it can be anything, or another cell. So it is eating. So that is called as a phagocytosis. The process is called as phagocytosis. If you are drinking something, it is called as pinocytosis. It has not been mentioned here, but write the name pino. Pino means pina. Pina is what? You are drinking something. So it is pinocytosis. And if it is degrading something which is present inside the cell only, that means it is possible that one of the endoplasmic reticulum has gone abnormal, or it has degraded, or it has become non-functional. Then also this lysozyme will be active, and it will eat this endoplasmic reticulum. But this eating is different from this eating. Why? Because here outside external factor is eaten, and here internal factor is eaten. So that is why it is called as autophagy. The term phagy is there, but auto means self. It is eating the self compartment. Okay, like autosomal diseases, self cells are degraded. That is why it is called as an autosomal disease. Okay, so it is autophagosome, autophagy. So what are the three different pathways? The first is the phagocytic pathway. The second is the pinocytic pathway, and the third is it is also called as an endocytic pathway. Okay, endocytic pathway, and the third is the autophagy pathway. Understood? These are the three pathways, and ultimately, see, they are mixing with this lysosome. They are mixing with this lysosome, and this lysosome is then breaking down the things. Okay, if you are talking about phagocytosis, suppose I'm talking about phagocytosis, there is one important receptor which is present, and it is called as the Clathrin receptor. When we are talking about the membrane receptors, we will deal that thing there, but just write down the name here, Clathrin. Can you see here, clip-like organs are present? This is called as the Clathrin. And what does this Clathrin do? It will hold off the foreign material. It will hold off, and it will then pinch off that thing from the membrane, and it will take the phagocytized material inside the cell. Okay, so once this phagocytic material has entered, your lysosome will become now active. Okay, I have to fuse, something has come inside. So it will go and fuse with this. This Clathrin coat will direct it to here, will direct it. Okay, and once they have been fused, now this Clathrin is very, very important for the for the holding of this. So this Clathrin will again be recycled, and it will again go to the plasma membrane for its other function, second time function. Okay, so what is the fate of this receptor? The question is asked for 2 marks. What is the fate of this Clathrin coated receptor? The fate is, it is recycled. Only in one line you have to remember, it is recycled, and it is not degraded. Even though it is merging with the lysosome, it is not degraded. Okay, it is not degraded. It is recycled. Very, very important, it is recycled. And from where these endosomes are coming? These endosomes are coming from your Golgi. From the Golgi, all the vesicles are budding off, and the vesicles with all the enzymes, they will fuse with this phagocytosed material. And when they are fusing with this phagocytosed material, it is called as a late endosome. Okay, a late endosome. And in the late endosome, what will happen? All the things will be degraded. After it has eaten up all the things, see the shape here. See the shape, see the shape. Okay, after it has eaten everything, it will come back to its normal shape. So if you are targeting this lysosome, you can see so many shapes. That is why it is called as a morphologically diverse. Okay, so this is your pathway. What was important here? The important thing here is the Clathrin coat. The Clathrin coats are very, very important. If you see this pinocytosis pathway, what will happen in this pinocytosis is this pinocytosis, there is no name of any receptor given. So there is pinching of this plasma membrane, and then this is called as a endocytosis. Okay, endocytic vesicle. It is called as endocytic vesicle. Okay, this endocytic vesicle will then fuse with the lysosome, and then it will be termed as your endosome. Okay, the endosome, where the lysosomes enzymes will act upon the pinocytosed material, and then it will degrade it. Okay, so these are the two pathways. You have to remember the pathways. What is the meaning of that pathway? Eating of solid particles is called as phagocytosis. Eating of the liquid particles is called as endocytosis or pinocytosis, and eating of the self parts or the autosomal parts is called as the autophagy. What is autophagy? The question will be asked like that. So you should know that what is autophagy. When self things are eaten. You have to remember this thing. The things which are present inside the cell only are eaten. It is not coming from outside. Understood, everyone? See, I was telling you about this phagocytosis, autophagy. So whatever I was telling you till now, that is written all here. Fine. Okay, we have discussed this. And see, one more thing you should remember, that what is this autophagosome? Okay, autophagosome. Sometimes this term is asked, that what is autophagosome? Autophagosome means the body which has engulfed the self compartments. When it is having the self compartment, suppose it has engulfed this mitochondria, with the presence of this self compartment only, it is called as autophagosome. So sometimes this term is called that, this term is asked, and you will be given 4 options, and you have to find out the right option, that what is autophagosome. Okay. Autophagy, just now I was telling you. Okay, now see here. The first thing which is happening is the formation of this phagophore. The second thing is autophagosome, which we were discussing just now. Okay, the phagophore, what is this phagophore? When there is like elongation of the membranes, when this mitochondria, protein, or peroxisomes, anything, when it is coming with the lysosome, there is extension of the membranes of the lysosome. So that first step is called as the phagophore. The first step. Okay, the first step is the phagophore. And when it has completely enclosed it with this, it has completely enclosed this is the name of the protein which help in the clipping off. Okay, in the clipping off, because how this lysosome will engulf, it has to open its pore. Okay, and once the pore are open, it has to close. Okay, so the first thing, when there is extension of the lysosome and all the particles are taken in, the first step is your phagophore. Mark these words. Okay, these words, sometimes they call they give you for definition. For 2 marks question they give you, and in 4 marks they will give you the steps, and they will ask you to rearrange the steps in proper order. Many times it is asked. So the first step is the phagophore. The second is when complete autophagosomes are formed. Then, when autophagosomes are formed, if it is fused with this lysosome, there will be degradation, and this degradation will lead to the complete breakdown of the engulfed material, and again your lysosomes will be released. Understood, everyone? So this is kind of a cargo. Okay, LC3B, just remember the name, from where it is coming, what is its function. That is not important. Just remember that it is one of the factor which is important in clipping of all these things, so that the lysosomes can bind very easily and it can give its function. Everyone got the point? This much only was there for lysosomes. And all the important questions I have discussed. From lysosomes, nothing will be asked out of this. Everything I have covered for lysosome. Okay, so from the next class, whatever are the small topics now, I will finish those things like the nucleus, the peroxisome, plastids, vacuoles. I'll cover all these topics, because this thing, endoplasmic reticulum, mitochondria, they will take time. They are very, very big topics, and they have so numerous function. So these are very big topics. And cytoskeleton also. It is a very big topic. I need, I think, 2-3 lectures to complete this. Okay. If I'm stretching it for 2 hours or 2.5 hours, only then I'll be able to complete in 2 days. So let's see that how we can do that. But definitely next class I'll be completing cell wall, nucleus, then peroxisome, plastids, and vacuoles, because these are small topic. Actually today only I was planning, but I'm busy with the workshop also. I have workshop at 7 o'clock. That is why I thought that I'll just complete this lysosome. Yes, 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 yes. Actually, not. Clathrin coat is important. Clathrin coat is important um, for receiving these particles which are present outside. For holding this particle. Okay, for holding these Clathrin coats are important. Okay, and what is the location of this Clathrin coat? Actually, the location of this Clathrin coat is on the membrane. But if this Clathrin coat is coming inside, so that is giving the signal to the lysosomes that Clathrin has come inside. That means some foreign particle has been engulfed and it has been taken inside the cell. So my function, now I have to do my job. It is like that. Okay, so Clathrin is not actually directing, but it is a kind of signal which is being given to this lysosome. Clathrin is important for holding this foreign particle so that it can hold, and then it can fuse it with the plasma membrane and it can bring it inside. Because do you think this bacteria and lysosomes, it can come to the hold of these cells? It is very difficult. So for that, they are having this Clathrin receptors which hold these particles. So important thing is not they are directing. Important is they are holding it and they are pinching it inside. They are making the particles force to inside. Forcefully, they are taking these particles inside. Okay, and sometimes what is happening is that our immune cells are also very active. So immune cells will target these viruses and everything. So when the immune cells are complex with this now, so something it should be there. They are bringing the immune particles and everything. They are bringing the antibodies and everything. They are bringing this particle to the cell. So there should be something which can hold off this. So these are the Clathrin coated pits which will hold off this debris or these nuisance particles, and then they will pinch it inside, because it is very important that they should be cleared, or otherwise what will happen, continuous immune reactions will be triggered and it will lead to an inflammatory situation. Sometimes you can see whenever there is some kind of infection which you are dealing, what will happen, there will be swelling, there will be redness, there will be pain. Why? Because continuous immune signaling is given. And when all this will be over, when all that debris are cleared. Okay, so these all things are giving the signal to the cell that okay, everything has been done, now clear off, clear, clear, clear. Because lysosome, what is the function? It is clearing. It is cleaning the debris. It is making the surrounding healthy and clean. Understood, Asha? Hope everyone is understanding and there is no confusion. This was a very small and easy topic, but very important topic. You saw that how many questions are asked, and we will solve the CSIR questions also, and you will see when I was talking about this acid hydrolases, pH 5, proton, all these questions have been asked. Understood? Any doubt you can ask me, or otherwise we will meet Thursday. Thursday, Friday, Saturday, fix 5 to 6:30 or 7. You have to bear with me. Okay? No doubts. Everyone, Deepshika, Mansa, you have understood? Priyama, both of you are not asking anything. That is why I am a little bit in doubt that you are getting the things or not. And how you will read this talk and okay. And how you'll read this, read these PPTs, you just have to make small notes. That's it, small notes. And I have told you that please buy Pathfinder. It is like Clathrin is a protein. Yes, it is a protein, Asha. Actually, it is a transmembrane protein which is like holding off a kind of receptor which is holding on this plasma membrane, and it is acting like clips. It is acting like the guards which have to look for all these debris particle, all these foreign particles. Okay. Everything most of the things now, they are proteins only. talk of enzymes, talk of this receptors. Most of them are proteins. Any other question? So see, we have completed this whole thing, and now we have completed this lysosome. And my promise is that I will complete the cell wall, nucleus, peroxisome, plastid, vacuoles, chloroplast. All this thing I'll complete on Thursday and Friday. 2 days I'll need. Okay. And one day I'll need for this mitochondria. Mitochondria 2 days, endoplasmic reticulum also 2 days I need. It is very big topic, very, very big topic. And this cytoskeleton, it is like vast topic. So I need time for that. It is a book, okay, Pathfinder. It is a book which has, which has organized all the notes now very precisely. So at the end, suppose if you are not having time to read, will it be possible to read so many PPTs at the end? It is not possible. Okay. And it is also not possible that you always make, because time is very, very important. So suppose if I am dealing this topic and now you have sat down and you are writing one point or like that, so it will take a lot of time. What you have to do is that you have to just jot down the important points, whatever the questions I am telling, whatever analysis portion we are doing, you just have to jot down that. And at the end, when you want to revise, at that time, this book will be a handy book to you. I completed it in like 1 year. 1 year it took me, but with the process which we are learning, you will take only 1 month to complete, and that also it will be like a revision for you. A complete book which is summarizing everything. Plus there are questions also, but I am solving all the questions for you. I'm giving you the questions from there, but at least you should have a reference book, because all these things you are given, you have been given, but at the end it is not at all advisable to read from the PPTs. Or if what is this possible that you can take the printouts of all these PPTs? No. And at the end there will be 100 lectures. It is possible that can you listen to all the 100 lectures? Yes, yes, it is available online, and it is of like 600 700 rupees. Two books are there, or you can get four books. There are four books pack. So if you want, you can take the four books. It is around 1,000 rupees, or you can take only two books, part one and part two. It is around 600 700 rupees maximum. Okay, so online it is available, on Amazon also, on Flipkart also it is available. Everyone, I'm telling you. Okay, everyone, please take this book, please. It's my request. And suppose if some faculties is not able to complete the portion, and if you're not satisfied with their performance, this book, the portions, you will be able to be justified. Okay, you can be confident when you are going in the exam. And if you have any doubts, there are other faculties. We have also cleared CSIR net, and we have also read the whole book, so we have the knowledge. But because it's a very vast portion, it is not possible for single faculty to complete it. Okay. Yes, you can go through, Asha. It is very good if you are revising each and every day. Yes, Pathfinder. Yes, right, Priyama, Pathfinder. If you have any doubt, anytime in any of the portion, you can anytime ask me. Pathfinder, just go through Pathfinder. That is ultimate book. Okay, just go through Pathfinder, because what I am doing is that I am taking it from multiple sites and I am giving you. Okay, some portions I am taking it multiple sites and I'm giving you. So you are getting the crux of each and everything. But at the end, when you want to revise now, read this book and everything will be in your mind. After the revision, suppose if you have bought this Pathfinder and I have completed this topic, lysosomes. Okay, so what you can do, you can go to that lysosome portion and you can read that portion and you can analyze that time. In just like 15 minutes you will complete that topic, and there are questions in behind that, so you can solve those questions. So it will be very easy for you, and your notes are already made. You have to save time. That is important. The important is revision. Okay, the important is you should get the concepts. The concepts is given by us. Okay, when you are clear with the concept, just revise it there. And questions we are discussing, just write those questions. That thing you have to do from our end, from when we are discussing the things. Okay, I'm giving you like a mantra. Okay, if you want to clear net in 6 months, and that also JRF. Okay, that also JRF. I got to know about these things very late, and this is from experience-based knowledge. I'm telling you. Fine, if you want to clear net, please do this. Fine, everyone. And if you are having any doubt in any of the portion, you can tell me any. For cell biology, carp is good. Carp and Cooper is very good. But the thing is that when you are going for the reference book, you will take a lot. Lodish is very good for cell biology. Lodish, Carp and Cooper are the best books for cell biology. But what is the problem? The problem is the description, the descriptive natures of those books. If I tell you immunology QB is the best book, but see the size of that book. It is 5,000 pages book. It is possible that you complete that book in 10 days. It is not possible. But I have read immunology multiple times because I am into this field like from 8 to 10 years. So I will read that QB and I will give you the steps. But what you have to do, you want to clear your competitive exam. That should be in your mind. So immunology is perfectly fitted and given in the Pathfinder in like 30 to 40 pages, and those 30 to 40 pages are enough to clear the exam. But definitely the concepts are not easy to understand. Okay, the concepts are not easy, because to read and to understand, when I started with the Pathfinder, I hardly completed one page in one day, and it's a truth. Okay, I'm not faking anything or exaggerating anything. But when I am making you understand, very easily you will get those things. Okay, so that book is like your for revision. Okay, revise the things from there, solve the questions from there. So at the end, when you are having 1 month, you will just make a plan. 1 week I will end up with this unit, 1 week this unit, 1 week this unit, like that you can do. But when you are just dependent upon this PPTs and everything, it will be difficult for you. Okay, because I know what is the limitation of the online classes. I know that. That is why I'm suggesting this to everyone. Okay, Anika?
Okay, thank you, ma'am.
So, okay. Thank you, everyone.
Okay, thank you. So, students, if you understood today's task, please comment yes in the comment box. Just let us know. Okay, thank you. Thank you, everyone, for your responses. Okay. Okay, now you can leave the meeting.